Right now, inside your body, something is happening. A cell is splitting in two. To become 4, 4 become 8. This is life doing what life does. Replicating, repairing, renewing. Your body carries out this process tens of millions of times every second. And the vast majority of the time, it goes perfectly. But occasionally, something goes wrong. And when it does, your body has defenses. Remarkable ones. So here is the question at the heart of this video. If your immune system is so precisely built to find and destroy rogue cells, then how does cancer ever survive at all? Cancer is not a foreign invader. It is not a virus, not a bacterium, not something that arrived from outside. Cancer originates from your own cells, your own DNA, behaving in a way it was never meant to. Every time a cell divides, it copies roughly 3 billion letters of genetic code after their own internal proofreading processes. The error rate drops to roughly 1 mistake per billion letters. But even at that accuracy, with tens of millions of divisions per second over a lifetime, mistakes accumulate. Most of them are harmless. Many are corrected automatically. But some mutations, in the wrong combination of genes, can trigger something that should not be possible. A cell that does not stop dividing. Now, here is what makes this so striking. Your immune system is specifically equipped to detect exactly this kind of cell. It does so continuously in all of us. In fact, researchers believe that cells with cancerous mutations arise regularly in the body, and that the immune system eliminates most of them before they develop into anything serious. So what changes when cancer wins? What does a tumor do? What does it become that allows it to survive, grow, and persist inside a body that was built to stop it? That is what we are going to explore. And the science behind it is more intricate than most people expect. At the core of your body's anti-cancer defenses is a system known as immune surveillance. The concept was first proposed by immunologist Frank Burnett in the 1950s and has since been supported by decades of experimental evidence. Your immune system does not only fight pathogens from outside, it also continuously scans your own cells from within, looking for anything that looks abnormal. Every healthy cell carries a kind of molecular display on its surface. These are called MHC, Class I molecules. Think of them as small identification windows that show a sample of what is happening inside the cell. If the cell is healthy, the display is unremarkable. If the cell is infected or undergoing abnormal changes, the display also changes. And your T cells and natural killer cells are highly sensitive to exactly these changes. Transplant recipients on long-term immunosuppressive drugs and people living with HIV who have low CD4 counts face a statistically higher risk of several cancers, not because suppression caused the cancer, but because the surveillance that was quietly preventing it has been reduced. A tumor that eventually grows into something clinically significant has, in most cases, already been through this gauntlet and survived it, not by chance alone. Through a process that took years, sometimes decades, to unfold. The relationship between cancer and the immune system is not a single event. It is a prolonged, shifting interaction. This process has a name. It's called cancer immunity. It was characterized in detail by immunologist Robert Schreiber and colleagues in the early 2000s. Immunoediting unfolds in 3 stages. The first is elimination. The immune system is actively winning. It detects and destroys abnormal cells. This is ongoing in all of us, throughout our lives. But occasionally, a cell survives, not because it is inherently stronger, because a mutation it carries happens to make it slightly less visible to immune detection. That cell replicates, and its offspring inherit the same advantage. The next generation of abnormal cells is, by the logic of natural selection, marginally harder for the immune system to recognize than the one before. This is evolution by natural selection, operating inside the body across years. The second stage is equilibrium. The immune system and the tumor reach a kind of standoff. Immune cells continue eliminating cancer cells, but cancer continues replicating. The tumor does not grow dramatically. It may remain clinically undetectable for years or even decades. Autopsy studies examining people who died of unrelated causes found that a notable proportion carried small, undiagnosed tumors that had never grown. Research has documented this particularly clearly for thyroid and prostate cancers. Their immune systems appear to have been holding those tumors in check. Silently, indefinitely. The critical question is not always whether those cells arise. It is whether they escape. A tumor that has reached the escape phase has not simply outrun the immune system. It has, through the accumulated pressure of evolutionary selection, developed specific mechanisms to neutralize it. To understand how, let us follow a single immune cell, a cytotoxic T cell, one that has been activated and is entering an established tumor with a clear purpose. This T cell has been primed. It carries the right receptor to recognize the cancer cells nearby. Under normal conditions, it would locate a cancer cell, make contact, and release specialized proteins, perforin and that puncture the cancer cell's membrane and trigger its controlled death. Now watch what the cancer cell does. The cancer cell is displaying a protein on its surface called PD-L1. This protein binds to a receptor on the T cell called PD-1, and when that binding happens, the T cell receives a signal. Not an attack, a message. Stand down. This is not the cancer cell destroying the T cell. It is the cancer cell sending a signal the immune system already recognizes it. PD-L1 and PD-L1 are part of a legitimate immune regulation pathway. It evolved to prevent T cells from attacking healthy tissue once an infection is cleared. It is sometimes called an immune checkpoint, a biological break. Cancer, through evolutionary selection, has arrived at a configuration that presses that break continuously. Cancer does not overpower the immune system through force. It uses the immune system's own communication language. It mimics the signals that mean, everything is fine here, nothing to act on. To be precise, cancer does not do this intentionally. There is no strategy or awareness involved. What we are describing is the result of natural selection. Cells that happen to express more PD-L1 were harder for T cells to kill. Those cells survived longer and replicated more. Over time, the tumor became dominated by cells with exactly this property. The outcome looks strategic. The mechanism is evolutionary. Some cancer cells progressively reduce the expression of MHC molecules on their surface, effectively removing the identification display that T cells use to detect them. Others secrete chemical signals that recruit a type of immune cell called a regulatory T cell. Regulatory T cells normally dampen immune responses. Inside the tumor, they suppress the very immune cells that should be attacking. Research published across major journals, including Nature and Cell, has identified dozens of these suppressive mechanisms. Immunologists describe the result with a specific term, T-cell exhaustion. T cells that have entered the tumor, encountered it, and gradually lost the functional capacity to kill, not because they are absent, because the environment has worn them down. Tumors actively reshape the environment around themselves. They secrete signals that stimulate the growth of new blood vessels, a process called angiogenesis. These vessels create regions of low oxygen, known as hypoxic zones. Cytotoxic T cells function significantly less effectively in low oxygen environments. The dense structural scaffolding that surrounds many solid tumors, known as the extracellular matrix, creates a physical barrier that immune cells struggle to penetrate in sufficient numbers. A tumor, in this sense, is not simply hiding from the immune system. It is actively remodeling its surroundings to make immune access harder. Now, here is where the story takes an unexpected turn. The immune system that cancer exploits so effectively is also one of the most powerful tools we now have to treat it. In 2010, a drug called ipilimumab was entering late-stage clinical trials for patients with advanced melanoma. Ipilimumab worked differently from any cancer drug that had come before. It did not target the tumor. It targeted the immune system. Specifically, it blocked a checkpoint protein called CTLA-4, and in doing so, it reactivated suppressed T cells. In a subset of patients, some individuals with advanced melanoma achieved sustained remissions lasting years. Response rates to immunotherapy vary significantly by cancer type, tumor profile, and individual biology. Many patients did not respond. The scientists whose research made it possible, James Allison and Tasuku Honda, were awarded the Nobel Prize in Physiology or Medicine in 2018. Rather than targeting the cancer directly, it targets the suppression. It removes the molecular brakes that the tumor has engaged. It restores the immune system's capacity to do what it was already trying to do. In cases where it works, the patient's immune system was already responding to the cancer. It was simply being held back. When that restraint was lifted, the immune response could resume. We are currently in one of the most significant periods of change in cancer medicine in decades. In roughly 15 years, immunotherapy has moved from a contested hypothesis to an approved treatment for multiple cancer types. Immunotherapy works well for some patients. For others, it produces little or no clinical benefit. The gap comes down, in large part, to the evolutionary history of the tumor itself. Through repeated rounds of immunoediting, some tumors have so completely lost their MHC surface displays that even a fully reactivated T cell cannot locate a usable target. Others have acquired suppressive mechanisms that the current checkpoint inhibitor drugs do not address, pathways that remain active even when PD-1 and CTLA-4 are blocked. The cancer continues to evolve in response to the treatment. New selective pressures drive new adaptations. There is no malice involved. No awareness, just the pressure of natural selection finding whatever survives. So, let us return to the question we opened with. If the immune system is so precisely built to find and destroy abnormal cells, how does cancer ever survive? The answer is not that cancer overpowers the immune system. It is that cancer, shaped by the pressure of immune attack, arrives at forms that the immune system struggles to act against. It reduces the signals the immune system reads. It activates the brakes the immune system uses. It reshapes the environment in which the immune system operates. All of this happens not through intent, but through the same mechanism that drives all biological adaptation, variation, selection, and inheritance. Cancer recruits immune cells as what we call it. It builds physical terrain that is difficult for immune cells to penetrate. It evolves continuously toward whatever configuration is hardest to eliminate. The boundary between the body and its diseases is not clean. Cancer does not arrive from outside. It emerges from within, from the same cells, the same genetic machinery, the same evolutionary drives, and the immune system that responds to it is not simply a weapon aimed at a foreign target. It is a system in conversation with its own tissue. There is something worth sitting with in all of this. We tend to speak of cancer as something external, something that attacks, something that arrives, but the research tells a more complicated story. Cancer is what happens when the cellular processes that sustain life lose the constraints that normally govern them. The immune system is what happens when the body has developed extraordinarily sophisticated ways of maintaining those constraints. Both are expressions of the same underlying biology. Right now, in your body, that process is active. Cells are dividing. Some are behaving abnormally. Proteins and immune cells are responding, correcting, resolving. For most people, most of the time, this happens invisibly and successfully. The body is not infallible, but it is extraordinarily capable, and understanding how it can be undermined is, perhaps, the most important step toward understanding how to help it.